Hello, good morning everyone. So today's lecture we are going to discuss about the general characterization of actinomytes. So in the previous lecture we have discussed about the fungi. What do you mean by fungi and what are the characteristics of fungi we have discussed. And we have also discussed about the classification of the fungi. म्हणजे फंजाय काय आहे, त्याचे जनरल कॅरेक्टर्स काय आहे, त्याचं क्लासिफिकेशन काय, या सर्व गोष्टीची माहिती आपण लास्ट लेक्चरमध्ये घेतली होती. So in the today's lecture we are going to discuss about a very important type of organism, that is actinomytes. So let's begin. What are the actinomytes? So these actinomytes, first of all you must know that actinomytes are a organism which looks like fungi but it is a bacteria. म्हणजे काय, हे बुरशीसारखा दिसतो ग्रोथ करताना पण तो आहे बॅक्टेरिया. ठीक आहे. So they are the gram positive, non-motile, non-spore forming, non-capsulated, contain muramic acid containing bacteria in the cell membrane of bacteria. So morphology which resembles like a fungi but it is a bacteria. So the cellular organization of typically this bacteria is a gram positive, and also these actinomytes are related to a mycobacteria and corynebacteria like. So these actinomytes are mostly present in the soil and which gives a moisty smell when there is a precipitation of rain. म्हणजे ज्यावेळेस जेव्हा पाऊस येते तर पावसामध्ये जे मातीचा वास जो एकदम सुगंधी एकदम खमंग एकदम वास जो येतोय तर तो वास हे ॲक्टिनोमाइट्समुळे, कारण दिस ॲक्टिनोमाइट्स प्रोड्युसेस सर्टन फ्लेवोनाइड्स कंपाऊंड and due to which this actinomytes gives a pleasant smell to this soil. So what is the role of these actinomytes? So the most of the actinomytes which are present in the soil which play a very essential role to fix the nitrogen and other compounds in the soil. म्हणजे काय, हे जे ॲक्टिनोमाइट्स आहेत तर हे ॲक्टिनोमाइट्स युजली सॉईलमध्ये प्रेझेंट असतात आणि हे वेगळंच दॅट इज सॉईलमधली जेवढी पण न्यूट्रिशनल व्हॅल्यू आहे त्याच्यामध्ये जे कार्बन आहे नायट्रोजन आहे यामध्ये पण फिक्सेशनसाठी या जे ऑर्गॅनिझम आहे याचा सर्वात महत्त्वाचा रोल प्ले करत आहे. तो जर त्याचं आपण क्लासिफिकेशन बघितलं, सो दीज ॲक्टिनोमाइट्स आर ब्रॉडली कॅटेगराइज डिपेंडिंग अपॉन द नीड ऑफ द ऑक्सिजन. दे आर ब्रॉडली कॅटेगराइज इन टू एरोबिक अँड anaerobic organism. So in an anaerobic organism the actinomytes are Rhotia, Biofibacteria, Arranchia, while the aerobic bacteria they are Nocardia, Actinomeriduna, Dermatophilus आणि Streptomyces. जर तुम्ही स्ट्रेप्टोमायसिस बद्दल तुम्ही जर व्यवस्थितपणे अभ्यास केला असेल तर हे बॅक्टेरिया सर्वात जास्त यूज केलं जातो अँटीबायोटिक्स प्रोडक्शनसाठी आणि हे इंडस्ट्रीमध्ये फार इम्पॉर्टंट आहे. आणि तो एक फंजाय नसून तो एक बॅक्टेरिया आहे, त्याची ग्रोथ पण फंजायसारखी दिसत आहे. सो दिस ॲक्टिनोमाइट्स वॉज डिस्कवर्ड बाय बॉलियर मॉल्ड. सो he name it is a ray like fungi and that is meaning that is a actinomytes. It is also isolated from various sources such as cattle and humans, and it is also isolated from the soils. The example of these actinomytes are A. Neosolidia, A. viscous, A. Odontocia, A. media, etc. So various there are examples, and so this most of the actinomytes are a pathogenic strain that is caused a chronic granulomatous infection. In certain way they are also used for the antibiotic production. So some are pathogenic, some actinomytes are the pathogenic which causes swelling in the connective tissue, and also when these actinomytes or certain strain which gives infection or which creates the infection in the humans which produces a sulfur like granules in the wound which are the specialized character or which are the specialized thing which is shown by the actinomytes when it causes the infection to human. So these actinomytes can also enter through the body by various sources such as from mouth, intestinal and vaginal opening. So this causes various infections, and this also This is also a very commonly causing organism which creates due to unhygienic nature of the human. When we discuss about the general characterization of these actinomytes, so they produce a minor streak that is 5 mm in size. Macroscopic bacteria, they are producing the colonies. The central, when we do the gram staining, is gives the gram positive nature, which is a mycelium clump which is formed in it, and the colony characterization shows a sun ray appearance. So in such a way, when we cultivate these actinomytes in a nutrient agar, it shows zone of inhibition also, and also a fungi like growth with a mycelium, small mycelium, and hence this actinomytes looks like a fungi. So in such a way, what do you mean by actinomytes, what are the morphological characters, and the classification and the example of the actinomytes we have discussed. So after completion this actinomytes, the next organism we are going to discuss about, that is a Rickettsia. So in that we will also discuss about the general features of Rickettsia. So the Rickettsia is a tiny non-motile gram negative bacteria, and the morphology is mostly pleomorphic in nature, that is it can change its shape. It is a bacteria not able to grow on artificial media. Also they are able to multiply in the cells of blood in the vertebrates, that is when we design a media which consists of blood contents, they show a rapidious or fastidious growth on that media. So this Rickettsia causes a disease which is well known related to Rickettsia, that is a Rocky Mountain spotted fever, that is it is caused by a tick. Then again the typhus fever from a leech, that is also Rickettsia. Rickettsia pox, that is caused by the bite of the mites, and scrub typhoid, this is also transmitted through mites. So in such a way, they are usually causing various diseases to human. So when we look over the culture of the Rickettsia, so for cultivation of the Rickettsia, mostly the two media are preferred for their growth, that is embryonic egg, that is yolk sac membrane, and next is a tissue cell culture, and in this way a Rickettsia can be cultivated. Next organism we are going to study, that is a mycoplasm. In the mycoplasm, it is also a bacteria which is facultative anaerobe as well as obligate anaerobe. It can be grown on artificial media, but the speciality of the mycoplasm is that it lacks the cell wall. So cell membrane is the outermost layer and it lacks the cell wall. This mycoplasm have a plasticity to show or many shape ranges from spheres to branching filaments. So this mycoplasm again not killed by penicillin, since the cell wall is not there, hence it does not show a appreciable response against the penicillin antibiotic. This mycoplasm has the genome about the half the size of bacteria, which ranges from 30,000 that is 30,000 nitrogenous base pair, and when they have able to synthesize in the colonies, when they are able to grow on a artificial media, it consists of mostly the fried egg appearance, that is their colonies show a half a fried egg like appearance when we grow it on a media. So we must know more about that. The mycoplasm is that this mycoplasm lacks the cell wall, and this mycoplasm when we grown, it is showing a semi transparent and a boiled fried egg appearance on the colony. So again, important characteristic of this mycoplasm is that they have a limited biosynthetic, that is biosynthesis ability, and hence they are unable to grow, mostly they are unable to frequently grown on various different media. In the sterilization which is related to mycoplasm, in most of the cases it can be easily killed by change in the pH or certain osmotic shock. It kills the mycoplasm. In such a way, we have discussed about the general characteristics of Rickettsia, actinomytes and mycoplasm. Thank you.